Well, thank you, Serena, for the introduction. Yeah, I'm working with the BC Ministry of Agriculture, and I'm trying my best to cover now in 20 minutes what has been my profession for more than 20 years. All right. Okay. Do you see it now?
Yep, there we go. Perfect.
Okay.
Thanks.
So if we separate the soil in its components, we have the mineral component. Then we have the water component. Should actually something happen now here. Okay, let's go back now. So we have the mineral component, we have the water component, we have air, and we have organic matter. And this is a little bit how I'm going to also structure this presentation before I touch on some soil health considerations. The mineral component is the backbone of the soil. The paramaterial plays a critical role for the soil development and nutrient availability. As a result of continental drift and several collisions with other land masses millions of years ago, BC is actually a big patchwork of minerals originated from all over the Pacific. The mineral component provides us with many nutrients essential to plant growth. At least this is how it started in soils. But once vegetation is established and organic matter has found its way into the soil, the recycling of minerals becomes more and more important as a source of nutrients. There are 17 essential plant nutrients. The latest in the family is nickel. Many textbooks still list only 16 essential plant nutrients. However, in the early 2000s, the first clear evidence of nickel deficiency was reported for pecan trees growing in sandy, poorly drained soils. So if you like to remember all the nutrients, here's the mnemonic, right? Actually, not all nutrients come from minerals. Carbon and oxygen comes from the atmosphere, and the hydrogen comes actually from the water. So I won't talk a lot about soil nutrients or pH. This will be covered in the following session by my colleagues. So just imagine you drop a sample of soil in the water. So particles settle down to the bottom, and the fraction or so-called separate class that settles first is sand. In a 15 centimeter high jar like this in the picture, sand will settle in a few seconds. In soil, sand provides us drainage and porosity. The separate class that settles next is the silt fraction. For this jar, it takes somewhere between 1 minute and 12 hours to settle out. Silt is very important for the plant available water in the soil. It is also the soil separate class in the soil that is most sensitive to erosion. The last separate class that settles in the clay is the clay fraction. So it can take weeks until it settles out. I will talk a little bit more about clay in the following slides. Together, they form what is called the soil texture. So in the soil, there are some very small particles that determine the physical and chemical properties of a soil, but also have a huge impact on living matter. They are called the soil colloids. Clay often consists of three-dimensional sheets of aluminosilicates. Often they are like the sheets of a book, right? There are also clays that are non-crystalline, but we don't worry about them. They are usually formed from volcanic ash. Clays have two unique and important properties. A, they are usually negative charged, at least in our part of the world. And second, they are very small. And together, they sum up to a huge surface. For example, if you take just 200 grams of loamy sand, the surface inside is about the size of a soccer field. And 99% of that comes actually from the clay particles. Another important class of non-organic soil colloids are iron and aluminum oxides. Some iron oxides, like goethite, for example, gives the soil this yellow brownish hue that we see. Hematite gives the typical red color that you may have seen in Florida or in Hawaii. So another colloid is the organic soil colloids, and I will talk about it in later slides. So in this picture, actually, I have two clays that are very frequent in BC. Morillonite clays tend to be predominant in the water lake deposits in the south, and illite in the south of the interior plains in the northeast of BC. So as a result of that large surface and negative charge, colloids can retain components that are positively charged, so-called cations. Many of our nutrients are cations, like calcium, magnesium, potassium, and so on. The property to hold and release cations is called the cation exchange capacity, and the exchange capacities, it also impacts, so it does not only impact the way of retaining nutrients, but also your buffer capacity. So the higher your cation exchange capacities, the slower your soil will acidify, but also the more lime you will need once you have to raise the pH. So water is essential for life. So we are now in the water section. Between 60 and 95% of plants consist of water. Water is also the habitat of most microorganisms in the soil, or actually all. Without the soil water, soil microbial activity will come to a standstill. On the other hand, too much water will reduce the activity of those organisms, especially those that need oxygen for respiration, including the roots. In the soil, there is always an equilibrium between nutrients released into the soil solution and nutrients on or in soil colloids. So, however, the plants also have, and together with their allies, the small microorganisms, they have strategies to obtain nutrients from the colloids that go beyond what we find in the soil solution. For example, the water in the rhizosphere around the roots, close to the plant roots, is often more acidic than the remaining pore space. Therefore, when we analyze soil water for nutrients or pH, it often does not reflect the true amount of nutrients available to the plants. This is why a soil laboratory often uses weak acids to extract nutrients or calcium chloride to determine the pH. So inside the soil matrix, there are pores of all sizes. Large pores, like those that we find in sand, drain readily. The water is not available because it drains. When drainage stops, we say the soil is at field capacity. On the other hand, on the spectrum, there are the very small pores. In those pores, water is so tightly attached to the soil that can't be extracted by the plants. Clay, for example, has lots of those very small pores. Therefore, it can hold a lot of water, but much of it is actually not available to the plants. When soil water is depleted, plants by plant uptake or evaporation, and it's impossible for the plants to extract it anymore, we say that the soil is at the permanent wilting point. The quantity of water between field capacity and the permanent wilting point is the plant available water. Silt, as I said, is a texture separate class that can hold the highest amount of plant available water. Organic matter can substantially increase it, and compaction can substantially decrease the plant available water in soil. So now if you think that the small pores are pretty much useless, no. They play an important role later on in the protection of soil organic matter, sometimes for thousands of years. From the previous slide, you may have seen that there are various forces on water, like gravity, capillarity, or metric forces. These are forces that attract water to particle surfaces, or osmotic forces, so salt forces. Therefore, tensiometers are often more appropriate to determine plant available water than measuring the total soil moisture content. Let's go to the air. There are distinct differences between soil, air, and the air in the atmosphere. So first, like humans, many soil organisms use oxygen and exhale carbon dioxide. Now, the circulation in the pores is not as good as in the atmosphere, so it doesn't move freely. No wonder it concentrates in the soil. Now, under anaerobic conditions, that means when oxygen becomes less and less available, either by flooding, compaction, or microbial activity, microorganisms that do not need oxygen for their respiration become dominant decomposers. Unfortunately, the metabolic byproduct from those organisms have often properties that are either toxic to plants or are pollutants to the environment. Nitrogen oxides and nitrous gas, for example, they form under mildly anaerobic conditions. As it becomes more anaerobics, more severe anaerobics, we notice methane and hydrogen sulfide. And under the most severe conditions, we actually find that we have ethylene. That is, the rings are here false. For nitrous gas and methane are very potent greenhouse gases, and hydrogen sulfide are even at lower concentration toxic to soils. to form those emissions, there are energy sources required. And therefore, soil that contain easily digestible organic matter is actually particularly susceptible to causing those emissions under anaerobic conditions. For the costs for ammonia are a little bit different. So the circle is here actually around ammonia. Here the PowerPoint presentation didn't really follow what I told them. So the release of ammonia is different. It happens when we apply fertilizer on dry and warm soils when the pH is pretty high. However, under certain environmental conditions, it can also occur in unmanaged soils. Okay, the presence or absence of oxygen determines what is called the redox potential. This is a property to reduce or oxidize elements. That means, in simple terms, to remove or provide oxygen. Besides the impact on the composition of soil air, as discussed in the previous slide, a soil's redox potential also impacts the availability and mobility of elements in the soil. Iron and manganese, for example, are mobile in the soil when reduced, so when there is no air, but they are immobile when they are oxidized. As a result, we see in soils that are underwater for a prolonged period of time bluish colors dominating because much of that iron is now reduced or even sometimes removed. You see it here and there. In layers of the soil where reduced and oxidized conditions alternate, we observe often red mottles or even bands as spectacular as you see in this picture, because oxidized iron concentrates in larger pores. The redox potential also impacts the availability of nutrients. Nitrogen and sulfur are more readily available to plants when oxidized, but are also more easily drained out of the soil. So what is organic matter? Soil organic matter refers to the carbon-based components of living organisms in the soil, to their metabolic products, but also the remains at various stages of decay. With other words, it consists of living organisms and dead material. Now, there are different schools. Some say we don't include living biomass, and others say we do. But since it's very difficult to actually separate that in analytical methods, most people stick to that definition. So you may have already, so let's go to the life. You may have already heard from soil trivia, there is more life under soil than on top of the soil, and that there in a single teaspoon, you find 1 billion bacteria, and you find several yards of fungal filament. But the most interesting part is actually that all of those organisms, we hardly know 1%. So the most obvious signs of life in soil are macroorganisms, such as the arthropods, right? Like millipedes, centipedes, billbugs, and so on, or architects like ants and termites. One very important group, which are sometimes almost invisible to the naked eye, are nematodes. They are plenty and everywhere. So just to give you an idea, I just quote here a famous nematologist. He said, if all matters in the universe except for the nematodes were swept away, our world would still be dimly recognizable. We would find mountaintops, valleys, rivers, lakes, and oceans recognizable by a thin film of nematodes. So nematodes are very popular bioindicators because they are so ubiquitous. And because they have a short generation, they are also very responsive to changes to the environment. They can be easily classified in trophic categories, basically based on what they eat by their mouth parts. Macroorganisms play an important role in shredding organic material. So they make it more available to the smaller organisms for decomposition. They also play an important part in mixing the material and stabilizing the aggregates, like earthworms do that. A somewhat less known function of macroorganisms is actually that they play a vital role in recycling nutrients. In fact, by consuming microorganisms or other macroorganisms, their contribution to the release of nutrients like nitrogen or for the carbon flux is crucial. Now let's move to the microscopic fellows. So microorganisms are strongly involved in the release of nutrients from organic matter. This is called mineralization. However, they also play an important role creating stable aggregates. They often produce gummy substances, and in the case of fungi, they have hyphae that help the soil to aggregate. Bacteria specialize more on the digestible parts, but they play also a very important role. For example, rhizobium can fix nitrogen, or bacteria like Nitrosomonas or Nitrobacter, they can actually transform ammonium to nitrate and nitrate, which is called nitrification. But unfortunately, under anaerobic conditions, there are also bacteria that go the other way, and they form actually nitrous gases, which is called denitrification. Fungi are the ones especially more on the complex components, but they too do not just decompose. A whole group of fungi called mycorrhizae are living in symbiosis with higher plants, who in return for sugar, they get then all the minerals and water. One group of mycorrhizae, the most important group, is contains or produces a glycoprotein called glomalin. It's a very resistant fungal glue that helps to stabilize soil aggregates. In soils that contain those fungi, and there are many soils, glomalin can make up to 20% of the soil organic carbon. So while most plants can actually form a symbiosis with mycorrhizae, there is one notable exception, which are the brassica that don't form mycorrhizae. So actinomycetes is another group I want to highlight. These are a special group of bacteria. They also specialize in decomposing more complex components, and you can see them sometimes on compost. Or I got a green card already here. Soil actinomycetes, like the fungus genus Frankia, can fix nitrogen, and they play an important part as a contributor to the nitrogen of natural soils in the Pacific Northwest. So let's for the non-living and non-tissue organic matter are very complex substances. And we distinguish here between non-humic and humic substances. Humic substances, here we have humic acids and fulvic acids. We differentiate them by their chemical composition, not so much by their chemical composition, but rather by their properties to dissolve in alkaline or acidic solvent. In the soil, we have two categories. One is the labile material. It has a half-life of a few weeks to a few years. It constitutes about 10 to 20% of the total soil organic matter. It often includes living matter, and therefore it is no surprise that the labile material is that fraction within the soil organic matter that provides readily accessible food. It also contributes to things like stability of aggregates and thus enhanced filtration. The passive organic matter, or stabilized organic matter, has a half-life of many, sometimes thousands of years. It makes more than 60% of the whole organic carbon. It's not so much the chemical recalcitrance that determines the duration, but more clay humus complexes or microaggregates that protect the soil organic matter. This is typically associated with properties like cation exchange capacity and water holding capacity. So here I just don't go to every single soil health indicator. There is a long list, and they have a couple of pros and cons, all of them, but one thing they have in common is that some of them are actually inherent soil properties and should not actually be called soil health indicators, like texture or slope or something like that. Then the lab analysis for some of those indicators is not trivial. It's pretty expensive, and not all labs can do it. And finally, we sometimes simply don't have enough data to actually know what we do with that. So the biggest weakness is the temporal and spatial variability of soil properties. So when you don't, if you don't know exactly the time you measure, you may have either, you come to totally different results. You see that on the top, if you compare two different soils, you may actually have the impression they're the same. And the other side, if you don't measure that long enough, you might actually not get the trend. Here's an article I found about research comparing the bacteria of fungi to bacteria to fungi ratio, and it showed that the ratio of bacteria to fungi is higher in the vineyard than the adjacent unmanaged area. In other words, there are more bacteria compared to fungi in a managed system in a field than in an unmanaged system. Not only, well, this is nothing new. We know that, right? So there is sometimes the research on soil health indicator gives us sometimes results we don't actually, either it is already known, or it doesn't really give us any clue on what we need that for in terms of management. Another example is there is a strong relationship between permanganate oxidizable carbon, which is a mouthful to describe active carbon, and total organic carbon. Now, we can actually easily calculate the total organic carbon from the soil organic matter just by dividing by 2. So, with other words, this is not in the sensational finding, right? In fact, an almost boilerplate conclusion of research on soil health that soil organic matter has a beneficial effect. That is, we hear that all the time, which begs the question is why we are sometimes spending that incremental, that money on fancy tests if there is no benefit or incremental benefit. Now, if you're still not convinced that organic matter benefits the soil, let me show pictures that perhaps tell you more than 1,000 years. So this is a project I was involved in many years. So the farmers in West Africa here grow their crop in soils that are almost pure sand and bare of any vegetation. The annual precipitation is seldom more than 600 millimeter, while the evapotranspiration can reach more than 10,000 millimeter. So what they do first, they dig a depression in the sand, then they fill the holes with organic matter, usually a mix of goat and sheep manure, sometimes cattle manure if available, sometimes even human soil. And this is the only amendment. Around this depression, they build mini dikes to keep water from running off. And in each depression, actually one kernel is placed. And then the farmers pray for water. And this is what it looks like at the end of a decent rain season, with only 4 components: mineral, water, air, and organic matter. So I hope I made it in time. I had to skip a couple of things, but yeah, I think I hope I kind of made my point.